Okay, folks, we're going to look at photosynthesis today, and there's a few slides I just want you to read through as review, and that's labeling the parts of the cell. Okay, so go through that. Go through the transport mechanisms, Science 10 review right there. And just a reminder, the photon is a packet of light, and it's broken up into the wavelengths, whereas red has got a large wavelength of 700 nanometers, blue has got a short wavelength of 400 nanometers. Okay, and photosynthesis in Science 10, you learned it that this was about converting solar energy into something that we can use in organic form of glucose, okay, that can then be used. And there's our formula, 666. So carbon, 6 waters, glucose. And we find this in plants, algae, and types of bacteria. Okay, quick identification here. Mesophyll layer in Science 10, you learn contain cells that are for photosynthesis, and these cells contain chloroplasts. And when a chloroplast hits, light is hit, green light is reflected to your eye. That means that the other wavelengths are absorbed in the 400 and 700 nanometer range of light. So chlorophyll is the light absorbing pigment, the green pigment. There's two types, chlorophyll A and chlorophyll B, that we're going to see. And there's also these things called accessory pigments that also support slightly different wavelengths of light. Interesting experiments. I break light into the Roy G. Biv of the spectrum, okay? And if I just screen it for green light, green passes right through chlorophyll, does not get absorbed, i.e. excite the molecules, and so we get almost 100% transference in the galvanometer. But if we now compare that to blue light, I allow blue light through, notice that almost all of it is being absorbed. And when it's being absorbed, that means that the chlorophyll molecules are getting excited. In the galvanometer, very low reading because almost all the light is absorbed. So chlorophyll A and B transfer at slightly different wavelengths. Chlorophyll A in the 400 nanometer range, 480 nanometer range for chlorophyll B, but then they get also excited down at about the 680 nanometer range. Yeah, at the red end of the spectrum as well. So two primary lights, red and blue. Okay, now when the chlorophyll gets degraded in the fall, other pigments come through. And we're going to see this in this chromatography lab we're going to do. And that's why we get these beautiful pigments. They're always there. It's just that the chlorophyll masks them. And when it dies in the fall, these other pigments come through. Very important, interesting experiment. Engelmann. He takes some algae. We know algae is photosynthetic, okay? And he shines light on the algae at these different spectrums, Roy, G, Biv. And what does he sprinkle around the algae is bacteria that need oxygen, aerobic. Well, when he shines light at these ends of the spectrums here, it obviously is making the most amount of oxygen because of photosynthesis, because look at all where the bacteria is. When he shines green light, no, no algae, or sorry, no bacteria are growing here because no oxygen is being produced. Go through this little video here. That will really help understand it, the process. Okay, so we've looked at chloroplasts. And we want to just take a little more closer look at inside a chloroplast, structure of a chloroplast. The outside part, this fluid-filled cavity, is called the stroma. The granum are stacks of thylakoid membranes, okay? And this is where the first stage, the light-dependent reaction occurs. And the second stage, the Calvin cycle, photosynthesis occurs in the stroma. So, a thylakoid is the first stage, the stroma is the second stage. Okay, the thylakoid membrane. Here's the membrane of the thylakoid, phospholipid bilayer. Embedded in it are these photosystems. Okay, with lots and lots of chlorophyll and accessory pigments that we looked at, embedded in them, right there. So if I was to draw one thylakoid inside it, it's called the lumen. Okay, so it's just another term, lumen. There's my thylakoid membrane, this thing right here. And then outside is called the stroma. So just give keywords, thylakoid membrane, lumen, and stroma. Energy molecules, we know about glucose. We made one the first week of class, carbohydrate. ATP, when ADP, diphosphate, gains a phosphorus, we call this phosphorylation. And when it gains it, now it forms 3 ATP. So this extra one makes it supercharged. Why? Well, remember what phosphate is from Science 10? I have got 3 phosphates now joined together. And that's going to make this kind of tail, we'll call it. There's my adenine. It's going to make it really supercharged. Look at all the negatives. I've got 9 negative charges now. Here, I've got 6 negative charges. And what do we know about negative? They want to get away from each other. So this is kind of like spring loaded. And when that pings off to form ADP, there's the phosphate. When it pings off, we release energy. Okay, the last one is something called nicotinamide adenine dinucleotide phosphate, and that's capable of gaining a hydrogen and 2 electrons to form NADPH, and we call that an electron carrier. So photosynthesis is a redox reaction. You learned about this in Science 10, that formula, but now we want to add to this. When carbon dioxide becomes reduced, Leo goes Gur. Gur is gain in electrons. So when it gains electrons and associated hydrogen ions, because positive, negative, we start making sugar. When water loses electrons, LEO, loss of electrons, Leo goes Gur, okay? We lose electrons, and in the process, we make oxygen.